Coming to the anterior lens epithelium, as you can see, it is located in the anterior part of the lens under the capsule, and they are cuboidal nucleated epithelium. Now they lie deep to the anterior capsule, extending up to the equator of the lens. Now it is more increased in the periphery of the lens. At the equator, they divide and elongating to form the lens fibers. So the density of the fibers are more at the periphery, and at the periphery these fibers or the anterior epithelia extend to or modify to form the lens fibers at the periphery. And this is that part of the lens that is metabolically active. Now the posterior end of the lens epithelium is absent actually because the cells are used in filling the central cavity. With regard to the different types of cells in the lens epithelium, it is divided into a central zone, an intermediate zone, and a germinative zone. So in the central zone, as you can see in this diagram, it is made up of central non-dividing cuboidal epithelial cells. Now the next zone is the paracentral zone that starts from this area to this. And then we have the germinative zone. The cells in this pre-germinative or in the intermediate zones, they are relatively smaller and cylindrical in this area. Coming to the germinative area zone, now here the epithelial cells are more columnar located at the main equatorial region, and these proliferate to form elongated fibers that run all around the posterior part of the lens. Coming to the lens fibers, we have already discussed that it is from the germinative anterior lens epithelium that the lens fibers arise. Now, when you cut these fibers, the cross section gives you a hexagonal shape to these fibers, and their formation occurs throughout life. right from the birth to the end of the patient's life. And this is what gives rise to the new structure of the nucleus and the cortex of the lens. Now, the fibers that are formed at the posterior end of the lens or that part of the lens fibers which are close to the posterior part of the capsule, these form the primary lens fibers. Those fibers which are seen at the periphery or in the initial stage, that is from the columnar epithelial cells, whatever starts to form is called as the secondary lens fibers. Now coming to the nucleus, we already know that it is the central part of the nucleus which contains the oldest fibers, and this is called as the embryonic nucleus. Now, depending upon the period of development, we are dividing it into an embryonic nucleus, fetal nucleus, infantile nucleus, and the adult nucleus. Embryonic, as you know, it is during 1 to the 3rd month of gestation. From the 3rd month of gestation to birth is the fetal nucleus. The infantile nucleus is from birth to puberty, and the adult nucleus is from puberty to the rest of life. So now this is the embryonic nucleus. Then you have the fetal nucleus around it. Then it is the infantile nucleus that forms around it. And finally, the adult nucleus that grows throughout life. And after that, you have the cortex, the anterior the capsule, all around it. Now coming to the cortex. Now cortex means we have a nucleus and we have the cortex, and we have what we call as the lens fibers. So we should know that it is the lens fibers which later form the cortex, and it is the cortex that later form the nucleus. So that's how it goes. Now they are located in the outer part or the periphery of the lens nucleus, and to be more precise, this is just outside the adult nucleus. And as I said, this is the youngest part of the lens fibers. Now, what are the differences in the fiber arrangement of the nucleus and the cortex? In the nucleus, the fibers are arranged in compact fashion, compact, tight fashion, because of which they are hard in their consistency, whereas they are loosely arranged in the cortex and they are soft in consistency. Coming to the clinical application of the nucleus, we should know that it appears, or the clinical appearance of changes in the nucleus and the cortex results in what we already know as cataract. With regard to the polar cataract, they are seen at the poles of the nucleus, poles of the crystalline lens, which appears as small opacities very adjacent to the lens capsules and the adjacent cortex at the anterior or the posterior poles of the lens. Capsular cataracts, they are actually small opacifications we see, and here the plane is different. This is mainly in the lens epithelium and the anterior lens capsule, which literally spares the cortex. Now there is a anterior capsular cataract and a posterior capsular cataract. So the anterior capsular cataract would be located anteriorly between the cortex and the capsule, and the posterior would be located between the posterior capsule and the cortex at the posterior end. Visually, the anterior capsular cataract wouldn't be causing much of a visual problem, because the light which comes from the distance is having a nodal point on the crystalline lens, which has a high contribution to visual vision perceived by the person. So it is the nucleus or the posterior pole of the crystalline lens which is the nodal point, and hence a cataract involving the posterior subcapsular area will affect both the distance vision and the near vision of a patient. In another way, normal cataracts only affect the distant vision, but if in a patient you see that his near vision is also poor, and if it's a cataract, it's usually gonna be a posterior subcapsular cataract. Now coming to the nuclear cataract. Nuclear cataract, you should know that it is actually the embryonic, that is the innermost nucleus, that is getting cataractus. Maybe it is the embryonic nucleus alone, or both embryonic as well as fetal, that may get the opacification. Usually the nuclear cataract is bilateral, and there are different types of cataracts in the nuclear cataract phase, that is cataracta centralis, lamellar cataract, sutural and axial cataract, total nuclear cataract. So actually we are discussing congenital cataracts, and in congenital cataract, first one was the polar cataract, next was the capsular cataract. Now the third one is nuclear cataract, and in that there are different layers of the nucleus that can get cataractus, as we have already discussed, and it is different. Each lamellae of the nucleus can get cataractus. Just the Y sutures that is present in the nucleus can get cataractus, or even the central part which is called as the axial cataract, can be seen in the nucleus, or the entire nucleus can get cataractus. The next is the lamellar cataract, which is the most common type of congenital cataract. And this is usually round central shell-like opacities which are seen surrounding the nucleus. More like only one single lamellae of the cortex seems to be involved in these types of cataracts. Like when you do a cross section, you happen to see that this nucleus is clear, whereas there is a white line that appears around the nuclear element, which is called as the lamellar or the zonular type of cataract. Next is the coronary cataract, or called as cataracta coronaria. Now this is actually club-shaped opacities. As you can see here, there are club-shaped opacities that come from the periphery and invaginates. This is called as coronary cataracts. They develop at the periphery of the cortex near the lens equator. And it is also significantly seen in large number of patients. Next is blue dot cataract. As the name suggests, there are a lot of blue dot like opacities which you can see in the usually in the cortical matter. Cataracta puncti ceruleae. These are opacities that are found in the as rounded bluish dots, and they lie again in the periphery of the adolescent nucleus and deeper layers of the cortex. To continue on with the ciliary zonules, the ciliary zonules are also known as the zonules of Zinn, and occasionally they are very fondly called as the suspensory ligaments of crystalline lens. Obviously, the ciliary zonules are transparent, they are definitely stiff and not elastic. They extend from ciliary body. This is very important. You should know that the zonules are starting from the ciliary body and runs all the way up to the lens equator circumferentially throughout. Its job is 1. to hold the lens in position, and 2. and help the lens in accommodation. With regard to the arrangement of zonular fibers, we should know that the zonular fibers arise from the posterior end of pars plana.
Collectively, these zonular fibers.
Now coming to the arrangement of zonular fibers, usually from pictures and depictions, so far we know that zonules are arising from the ciliary body. But one thing we have to be sure about here is it is not arising from pars placata as you have seen in these pictures. It's in fact actually arising from the posterior end of pars plana, that is from here, and it is going all the way up to the crystalline lens. So it is arising from the posterior end of pars plana. This is the anterior end, this is the posterior end of pars plana, and this is located around 1.5 millimeters from the ora serrata, which is the anterior most end of the vitreous body. So if this is ora serrata, the origin of the zonules is 1.5 millimeters and arises from the posterior end of pars plana, and from here it drives all the way passing the pars plicata and gets attached to the equator of the lens. Now with regard to the zonular complex, that is the zones of the zonu or suspensory ligament is actually divided into pars orbicularis. Now this is the area that passes forward over the pars plana from its origin. Now here at this particular point, these have segmented into zonular complexes. As they pass through these folds in the pars plicata, they have divided themselves into zones or zonular plexus. From pars orbicularis to zonular plexus, and it goes to form the zonular fork. In the zonular fork, what happens is the zonules, after passing through the zonular plexus, they consolidate together and they cross around each other at the zonular fork, and then they move over the crystalline lens. So this is the zonular fork area. And this crisscrossing is at 90-degree angles. Now finally the zonular limb is actually 3 in number. So the anterior is the zonular limbs or the orbiculo-anterior capsular limbs. The next is the equatorial zonular limbs. Those are the zonules that are attached to the equatorial region. And thirdly are the posterior zonular limbs which are attached to the posterior capsule. So there are 3 layers to this as zonular limbs. One attaches to the anterior capsule, the next to the equator, and third layer attaches to the posterior capsule. One thing I would like to highlight here is in this diagram it's very clearly evident about the sutures which you see in the lens nucleus. You should know that the Y sutures which we see in the nucleus there is an anterior Y suture and a posterior Y suture. As you can see, it is the anterior one that is in normal orientation, and the posterior one is a inverted Y suture. With regard to the insertion of the zonular fibers, we know that it is inserted anteriorly and posteriorly at the periphery and at the equator of the lens. The layer of inserting zonular fibers and the related capsular layers are called as the zonular lamellae. And as we have already mentioned before, the zonules are non-elastic. The zonular lamellae are less compact. They are rich in glycosaminoglycans than the rest of the capsules, and the lamellae contributes to zonular adhesive mechanisms. It is a pericapsular membrane. With regard to the zonules, the clinical application stands as ectopia lentus, which means there is actually a displacement of the lens from its normal position. It can occur congenitally, developmentally, or acquired because of trauma. It is called as dislocated when it has moved away from its place. It is subluxated if it is maintaining the plane of the crystalline lens but is decentered inferiorly, superiorly, medially, or nasally. So, if a crystalline lens has displaced from its vitreous and has fallen into the vitreous or has come anteriorly into the anterior chamber, then we call it as dislocated lens. If it is in its own plane but is a little deviated down, up, nasal, or temporal, then it is called to be subluxated. So, subluxation is actually a partially displaced from the normal position, but it remains in the pupillary area, whereas dislocation means it is a complete displacement from the pupillary area and separation of all zonular attachments is a main reason. Ectopia lentus, the causes are very important. Developmentally, it can occur because deficiency in the development of zonule, which causes an ectopia lentus, and this is associated with decreased vision, marked astigmatism, monocular diplopia, and moving or shaking of the iris called as iridodonosis. Acquired lens displacement mainly occurs because of trauma. This is a very important slide. The ectopia lentis is associated with various syndromes, that is Marfan syndrome, homocystinuria, and Weil-Marchesani syndrome. So, the direction of displacement of the lens is again very important. It displaces upward and temporal in Marfan's, that is, it is superotemporal displacement. In homocystinuria, it is entirely opposite. It goes downward and nasal in homocystinuria. Weil-Marchesani, it can be in any direction. Usually it is forward subluxation of the crystalline lens. Now coming to the last segment of the lens, that is the biochemical part of lens, you should know that it is composed of 60%, 65% of water, and the next best content is protein, and it is this denaturation of this protein that results in cataract. Now the other things that we find in the lens is lipids, glucose, etc. With regard to water, which is 65%, 80% is in free and 20% is in the bound form. It is actually present in a dehydrated state that is maintained by the sodium potassium pump, just like in the stroma of cornea, which maintains the transparency of the cornea. Again, here also it is the sodium pump that plays important role in maintaining the lens transparency and the refractive index. With relation to the nucleus, cortex is more hydrated than the nucleus. The different type of proteins that you see in the lens, it is higher than that of any other organ in the body. The lens has the highest concentration of protein in the body. It is divided into two major groups. One is insoluble and soluble. The insoluble is albuminoids and the soluble ones are crystallines. Among that, albuminoids form only 12.1%, whereas crystallines are further divided into alpha, beta and gamma crystallines. And in that it is the beta crystalline that is seen at higher concentration. Now, what are the role of crystallines in the lens? It helps in the refractive property of the lens. The change in shape observed during differentiation of an epithelial cell into a lens fiber is again helped by the crystallines. It provides the lens with a stress resistant property and it has a chaperone like function. Chaperone is actually someone who looks after an accompanying person. So crystallines take care of the lens in its well-being. It is also having a role in the hardness of the lens. This is a small table with regard to the amino acids. There are two forms, proteogenic and non-proteogenic. They are actively transported into the lens. With regard to glucose, it is actually 1/10 of that what you find in the aqueous. There is also fructose, glycogen, sorbitol and inositol. So aqueous is having higher concentration of glucose than the lens. Lipids are 2.5%. Coming to the electrolytes, we should know that potassium is more within the lens. So potassium is more within the lens. So obviously sodium is more in the aqueous. Sodium is relatively lesser, calcium is 0.4, anions are like chloride, bicarbonate, phosphates are also present. Glutathione is present in the lens which is around 3.5 to 5.5. Ascorbic acid is another important content of the crystalline lens that is around 30 milligrams per 100 gram weight of the lens. Now the metabolic activities of the lens, the metabolism, the major site is in the epithelium and lens requires a continuous supply of energy for active transport of ions and amino acids, maintenance of lens dehydration and thereby the transparency, for the continuous protein and GSS synthesis. Most of the energy that is produced is utilized in the epithelium, which is the major site of all active transport processes in the lens. The sources of nutrient supply. Being an avascular structure, the lens takes nutrients from two sources of diffusion, that is from the aqueous humor that is anteriorly located and which is the main source, and also partially from the vitreous humor. The glucose metabolism, just like in cornea, it is the main source of energy. It enters the lens from aqueous and vitreous through simple diffusion and is metabolized through four main pathways. There is anaerobic glycolysis, Krebs cycle, and a Champion pathway. Because both sorbitol and fructose have the potential to increase osmotic pressure, it causes water to enter cells, and these sugars may help regulate the volume of lens. Now these are the different pathways that you see in the lens. There is anaerobic glycolysis and the energy produced is 2. There is Krebs cycle that gives around 36 ATP. HMP shunt pathway nil. Sorbitol pathway gives 2 molecules of ATP. The protein metabolism, it is synthesized from amino acids which are actively transported into the lens from aqueous. Synthesis of protein is slowest in the nucleus, and protein breakdown is catalyzed by peptidases and proteases. The transport mechanism to provide nutrients for metabolism, regulates water and cation balances in the lens, it disposes waste products of metabolism. The two transport mechanisms available are the active transport and passive transport. The sodium potassium is all coming in the active transport. Passive transport is mainly taken care of water, lactic acid. Now this is a very important diagram back again. So it is actually comparing the amount of various ions in the aqueous humor as well as the lens. As we have already discussed, the amount of potassium is high in the lens and sodium is higher in the aqueous humor. The content of water is 99% in aqueous humor and lens is relatively dehydrated to maintain the transparency at 66%. You should know that chlorine is higher in the aqueous, and the another important thing is the amino acids which are more in the lens. The transport of amino acids and inositols, they are pumped into the lens through anterior capsule. In addition, lens can also convert keto acids into amino acids. Glucose transport is by simple and facilitated diffusion across the anterior and posterior surface of the lens. So, coming to the lens transparency, we should note that the factors which play a significant role in maintaining the clarity and transparency are the thin epithelium, regular arrangement of the lens fibers, cellular organelles are very minimal and there is little extracellular space, and there is a lamellar conformation of lens proteins. Along with that, the relative dehydration of the lens helps, the semi-permeable character of the lens capsule, avascularity, auto-oxidation and pump mechanisms all have important and major roles.